Hello everyone and welcome to today's presentation. Rapid RGR-dependent visual pigment recycling is mediated by the RPE and specialized Müller glia, presented by Dr. Aleksander Tworak from the Center for Translational Vision Research at the University of California, Irvine, Gavin Herbert Eye Institute. My name is Stefan Pellenz and I will be moderating this webinar. I'm a product manager at Antibodies-online and I coordinate Antibodies-online customs validation program. This webinar is being recorded and we will make it available on our website within the next week. Any personal information about participants will be removed, so don't hesitate to use the chat window to ask any questions that will be answered in the Q&A session subsequently to the presentation. Organizer of today's webinar is Antibodies-online. Antibodies-online was founded in 2006 as a spin-off of Aachen Technical University in Germany. We are now located in Aachen, Germany, as well as in Pennsylvania, in Philadelphia more specifically. And we are offering more than 4 million proteomics products and from approximately 250 suppliers. We considered our mission to make the search and purchase of antibodies, proteins, kits, and other proteomics products easy and straightforward. Today, we count more than 6,000 institutions in 65 countries as our customers, some of which are indicated here. In 2013, Antibodies-online launched the Independent Validation Initiative. This is our approach to validate the proper function of antibodies, ELISA kits, and other products from our catalog under real-world conditions to assure their reproducibility. In 2015, we extended this initiative to our customers. So through our customer validation program, we offer a full refund for life science products sold through our platform in exchange for a validation report. Up to today, we have concluded more than 1,000 validations of almost 900 of our products in collaboration with more than 200 scientists. In case you're interested in validating any of our products in your particular experimental setting, please feel free to get in touch with us at independent-validation@antibodies-online.com. And this is actually how we got into contact with today's speaker, which is Aleksander Tworak. We are happy to welcome Dr. Tworak from the Center of Translational Vision Research at the UC Irvine. Dr. Tworak did his undergraduate research work at Adam Mickiewicz University in Poznan, Poland, and obtained his Ph.D. in biotechnology in 2014 from the Institute of Bioorganic Chemistry at the Polish Academy of Sciences. He was working for this work on microRNA biogenesis as well as the formation of RNA-G quadruplexes. During his postdoctoral training in Dr. Krzysztof Palczewski's lab at Case Western Reserve University in Cleveland, Ohio, and later then UC Irvine, Dr. Tworak switched his research to focus more on ophthalmology. More specifically, he studied the phagocytosis of photoreceptor outer segments and regeneration of the visual pigment component 11-cis retinal in cells in the retinal pigment epithelium, RPE. As an assistant project scientist, he is now working since 2020 to elucidate the role of the retinal G protein coupled receptor, or RGR, photoisomerase activity in the vertebrate eye. Dr. Tworak's work has been published in numerous peer-reviewed journal articles, and part of that he will be presenting in today's webinar. So with this short introduction concluded, I'm happy to give the floor to you. Take it away, Dr. Tworak.
Thank you very much, Stefan, for this nice introduction. And I'll try to share my screen now. Can you see it?
Yes. Seems to be working perfectly fine.
Great. Yeah, so welcome everyone. As Stefan mentioned, the title of today's talk will be Rapid RGR-dependent visual pigment recycling is mediated by the RPE and specialized Müller glia. And okay, let's start. So I'll give you a short introduction into the eye biology. So this is basic. Maybe let me just turn on the pointer really quick. Okay, perfect. Can you see the pointer?
Yes.
I hope it's perfect. Yeah, so this is the schematic of the structure of the eye. And as you see here in this panel B, the retina, which is here in the yellow layer in the back of the eye, has this nice laminar structure where half of the thickness of the retina is actually occupied by the photoreceptor cells. So the neurons that are the primary sensors of the light. And in principle, we have two major photoreceptor types in mammalian eyes responsible for vision forming, for image forming vision. And these are rods that operate mostly in low light conditions and exhibit fast saturation, and cones, which are primarily used in the high light or intense light situations, and their responses therefore do not saturate that fast. So in principle, in both photoreceptor types, the vision starts when the light hits the opsin protein that is conjugated with a so-called visual chromophore. It's always 11-cis-retinal, and the absorption of the photon causes the isomerization of 11-cis-retinal bound to the opsin protein to the all-trans form, which triggers further steps in the vision process, so the signaling through G proteins and so on, that initiates the visual perception. Subsequently, all-trans-retinal is released, and to sustain and maintain continuous vision, the all-trans-retinal needs to be re-isomerized to the 11-cis form in order to provide continuous supply of this photoactive form of the opsin, either rhodopsin in rods or cone opsins in cones bound to 11-cis-retinal. So in principle, we know two enzymes in the eye that are capable of catalyzing the trans-cis re-isomerization of the chromophore, which is basically required for the regeneration of this visual pigment, which is 11-cis-retinal opsin. So the first, so the most studied and the first recognized one is RPE65 protein, so retinoid isomerase that was established to be a crucial part of the so-called classical visual cycle. So this is a cycle that involves also other enzymes like LRAD and some retinoid binding proteins that allows a regeneration of 11-cis-retinal in all mammalian eyes. The issue with this cycle is that it, through a lot of experiments with electrophysiology, with biochemistry, it was established that this cycle itself is not efficient enough to sustain continuous vision under bright light conditions when the cones are actually the photoreceptors that provide or that initiate the visual perception. For that reason, for many years, other isomerases were studied whether they have importance for the eye. Some of them were initially proposed to have some importance, to have the trans-cis isomerase activity. Later, maybe this was disproved. However, what we know now is that there is at least one additional protein known as retinal G protein coupled receptor, so RGR, in the major topic of my today's story, that also possesses this trans-cis isomerase activity. And this activity is a part what we call now the photic visual cycle. And the reason is, I will touch base in a minute about this, that actually this protein is a photoisomerase, so its activity depends on the presence of light as well. And in principle, as you see here, the graph that I'm displaying is pretty complex. And the reason is not that the process is complex itself, but we are still not quite sure about what are the exact intermediates that are formed throughout this process when the photic visual cycle is operating. However, we know that the crucial part, so the activity of RGR, is dependent on light and is present in both RPE cells, so the cells that line up the retina from the back of the eye, and the Müller glia cells, so the cells that actually penetrate almost the entire depth of the neural retina in also all mammalian eyes. So let me just very quickly summarize what is believed to be the importance of what is the important aspects of this photic visual cycle, or what's the background for its importance. So first of all, despite the similarity in pigment concentration, density, and bleaching levels upon bright light exposure, cones were shown to restore their sensitivity about 10 times faster than rods. And this is interesting because it suggests that there is some additional or privileged supply of 11-cis retinals or of the visual pigment to cones under the bright light conditions. And for sure, the classical visual cycle, so the RPE65 dependent one, doesn't offer such feature to cones. So that's why it was believed that there must be additional cycle involved in regeneration of the visual chromophore. Next, the RPE-based classical visual cycle, which is so far the best characterized pathway of all-trans-retinal isomerization to the 11-cis form, is too slow to meet, as I mentioned, is too slow to meet the chromophore regeneration demand of the cones under sustained light conditions. And the third important aspect is that the RGR-dependent photic cycle is so far the only prominent alternative source of trans-cis isomerase activity in the retina known to date. So this last point basically explains why our lab was interested in exploring the importance of RGR for the vision and for the regeneration of visual chromophore. So what is RGR? What is the activity of RGR? As I mentioned, RGR is a photoisomerase, so it binds all-trans-retinal and it converts it to 11-cis upon the stimulation of light, and it does it most efficiently when the light is in the range of green wavelengths, so at around 500 to 530 nanometers, as was shown in our previous study published in 2019. And we also showed in this study that additional protein, retinoid binding protein known as CRLBP, strongly enhances the generation of 11-cis retinal by RGR. So as you see here, the experimental setup in our experimental setup, this was the level that RGR was able, that 11-cis retinal was generated upon just the addition of RGR. Addition of CRLBP was boosting in the same conditions the generation of 11-cis retinal by 4-fold or so. And in the same study, we also established that a particular amino acid in the structure of RGR, which is the lysine 255 in helix 7, is responsible, is kind of a not a catalytic core, but the activity site of the enzyme. So the enzyme, so the site where the so-called sheaf-based formation occurs between the retinoid moiety and the protein that allows that precedes the photoisomerization process. One curiosity that we observed in our 2019 study was actually the distribution of RGR among different mammalian retinas. So observed that in human and also bovine eyes, RGR is fairly expressed at fairly similar levels in the RPE cells, so the cells that line up from the back the eye cup, as well as in the Müller glia. However, in mouse, so the most common animal model used in the laboratory, the differences between the expression of RGR between RPE and Müller glia are very high, and Müller glia seemed to express much less RGR. And this was kind of a observation that we didn't pursue any further at that time. However, in my today's talk, I will be addressing several issues, including this one that I just mentioned. Namely, I will be focusing on answering the question, what is the capacity of RGR-mediated photic visual cycle to supply 11-cis-retinal in the context that whether it's sufficient and effective enough to bridge this gap between what is produced by the classical visual cycle and what is the demand in the high or intense light conditions. What is the distribution of RGR across mammalian retinas? So this question stems from this observation that I just mentioned, that the distribution seems to differ between different animal species, mammalian species. And we are also interested because RGR, as I showed you, is expressed exclusively in two cell types in the retina, so the Müller glial cells and the retinal pigment epithelial cells. So because these are two distinct pools of cells supplying presumably 11-cis-retinal to a photoreceptor, so we were wondering whether any of those has more or less influence on the vision, or what is the relative contribution of RGR pools originating from both cell types to human vision or to mammalian vision in general. Okay, so let's start with looking into our studies on the first question. So what is the capacity of RGR-mediated photic visual cycle to supply 11-cis-retinal? So recently in our lab, we developed a nice method. Okay, let me start, maybe let me just take one step back. So the photoisomerization of RGR actually involves two very distinct steps. And the first one is basically the binding of the retinoid moiety by the protein. Next, the actual photoisomerization occurs. So by photoisomerization, we mean the isomerization of all-trans-retinal to the 11-cis form. And the next step is actually the release of the isomerized retinoid from this sheaf base, so from the state when it's bound by the protein to the surrounding membranes. And this is probably followed by the pickup of this protein by the CRLBP protein, because as I mentioned to you, as I showed in the introduction, CRLBP is facilitating the activity, and we believe that this is because it's actually picking up the product very quickly. It has high affinity to 11-cis-retinal, and also it's protecting it from re-isomerization back to the old trans form. So we know that the photoisomerization is a very fast process occurring in the range of femtoseconds, but this release is actually the step which limits the effectiveness and the productivity of this cycle. And for that, to study the kinetics of this release, our lab developed a brand new method, which I'm not gonna discuss in detail, but I'm referring you instead to this paper in PNAS in 2022 by John Hong. In principle, this method allows us to study the release of the retinoid from the opsin proteins in native conditions, so in native membranes, using a combination of chromatography and mass spectrometry. And what we observed was that the rate of hydrolysis of 11-cis retinylidene sheaf base, so the 11-cis retinal bound to the opsin, in this case to the photoactivated RGR, was on the order of seconds, with the half-life of 7.5 seconds in 20 degrees. And it's important to put it in a context. So in this paper that I mentioned in PNAS, we established the hydrolysis rate of the photoactivated rhodopsin. So the opsin that actually utilizes 11-cis retinal at the beginning of the visual process. Oh, okay, so I have the scheme here. So now we are basically comparing the rate of production by RGR of 11-cis retinal to the rate of the release of all-trans-retinal from the photoactivated opsin protein. Yeah. And as you see here, RGR is capable of producing 11-cis retinal much faster than the release of all-trans-retinal occurs in case of rhodopsin protein, which suggests that at least in this comparison, RGR is fully capable of supplying more 11-cis retinal than is needed, considering how fast the 11-cis retinal is utilized by the rhodopsin protein. So we observed, and we know now that RGR-mediated photic visual cycle is for sure capable of significant 11-cis retinal production. Now addressing the second question of the which I stated at the beginning, what is the distribution of the RGR protein among mammalian retinas? So we turned to single-cell RNA-seq data to study a little bit more this phenomenon. And what we observed, in this study, we tested the single-cell RNA-seq data from 3 different species, from mouse, macaque, and human. And despite some technical variations between these approaches, we were able to reliably identify decent, significant pools of Müller glia. And among those, only a fraction was always showing the expression of the RGR protein. Curiously, however, the fraction in, for example, macaque was on the order of 7–8%, while the fraction of Müller glia expressing RGR in human retinas based on single-cell RNA-seq data was quite, or the ratio between the proteins that were expressing or were actually showing no expression of RGR was quite the opposite. So we observed almost 96% of RGR, of Müller glial cells expressing RGR. So this is an interesting observation, and we don't have good explanation for that, and this for sure warrants that further studies are needed. But interesting observation by itself is that there's only a fraction of Müller glia that actually express RGR protein in all mammalian retinas that we tested so far. Yeah, so we took advantage of this observation, and we actually compared the transcriptomic profiles of the Müller glia that were expressing RGR with the Müller glia that were not expressing RGR. So we ran the classical RNA-seq analysis where we looked for differentially expressed genes between those 2 pools of Müller glia, and then we ran analysis to establish the overrepresentation of certain biological terms in this pool. And you see the result of this analysis here, the so-called gene ontology terms from the category cellular component by biological process are shown here, the most enriched ones. And as you see, when it comes to the cellular component, the most prominent term that was enriched in this comparison was the extracellular exosome. And in principle, the release of exosomes you know, this aspect of the biology of cells is now gaining more and more attention, and this result basically suggests that maybe the RGR positive Müller glia have some higher release of exosomes, but we have no idea how this is exactly related to the biology of those Müller glia and the photic visual cycle that they mediate. However, quite interesting observations came also from this second analysis, so the biological process GO-terms analysis. So here, the highest enriched GO-terms, several of them were directly related with the retinal biology, so retinal homeostasis and visual perception here. So if we look closer at the genes that were actually driving this overrepresentation and enrichment in this comparison, we saw here in this visual perception category, two very interesting proteins popped up. So the first one is RLBP1, which is the code name for the gene that encodes CRLBP. So in principle, this observation suggests that the level of CRLBP in Müller glia is somehow correlated with the level of RGR in those cells, which would again confirm its important involvement in processivity of the photic visual cycle in those Müller glial cells. And another interesting observation is that actually enriched in this pool was also one of the retinal dehydrogenases known as RDH11. And I have a comparison here which shows you the expression of all major RDH proteins in the macaque retinas. Also, this analysis was done on macaque data because they presented the highest quality, and also the highest number of Müller glia was unidentified in this, or the much higher number than in mouse. We were able to, much higher number of transcriptomes was analyzed in this analysis, the mouse transcriptomes. We did not get as many to perform such a detailed analysis. So in this macaque, what we see is that in the Müller glia, actually RDH11 is the most prominent RDH enzyme expressed in both foveal and peripheral parts of the retina. So the biological role of RDH11 is not very well established, but what is known for sure is that those RDH enzymes exhibit some level of redundancy, so that if one is missing, the other can take over its function. There's a lot of them, as you know, indicated by this number RDH11. I think it's up to RDH14. Not all are expressed in the retina, but several of them are. So this would indicate that not only are the RGR positive Müller glia are correlating the expression of RGR with CRLBP, but also with RDH enzyme that is, as we believe, important for the processivity of this photic visual cycle. So our conclusion from this observation is that it's likely that Müller glia exhibit some level of specialization within some subpopulations in the support of photic visual pigment recycling and photic visual cycle. So it is known that the presence of CRLBP is known to strongly promote production of RGR, as I mentioned, likely by the protection of chromophore from loss and from the thermal reisomerization. And RDH11 demonstrates equal specificity towards both cis and all-trans-retinoids. This is some information important considering the involvement of this RDH11 in the photic visual cycle. And Another interesting observation was that our RGR positive cells were also showing elevated expression of major retinal glucose transporter known as GLUT1. And this observation, on the other hand, could reflect the dependence of this photic cycle on some source of energy, and in this case, it would be the glucose. This is, however, very far-fetched theory about all those involvement of all those proteins in support of RGR, and of course, many more studies are needed to very well establish their role. So let me just now move forward to the third aspect of our study. So here we were, as maybe you remember, we were interested in establishing the role of the two pools of RGR, so the RPE pool and the Müller glia pool, on the support of vision, yeah, so on the regeneration of the visual pigment. And for that purpose, we actually developed a specialized mouse line. We called it RGR stop. So basically, we introduced a stop cassette between the exons 1 and 2 in the RGR gene that was accessible using Curie recombinase. So using this mouse model, we aimed to develop mouse lines that would be able to express RGR only in one of those two specific cell types of interest. So let me show you, I think. Okay, I'll come back to in a minute to that. So in the baseline situation, this mouse is essentially a knockout of RGR, and we confirmed that using immunochemical assay. And what we observed, just by comparison of the wild type and the knockout animal, was that the loss of RGR doesn't really affect the structure of the retina. Yeah? So we didn't observe any signs of retina degeneration or other structural issues in the retinas as old as 12 months old. However, what we observed in those animals was that by comparison of the levels of different proteins involved in retinoid metabolism, we saw that actually one protein, was very highly upregulated upon the knockout or the loss of RGR, and it was the LRAD, so the important enzyme involved in this classical visual cycle, so the major source of 11-cis retinoid in mammalian retinas. So this was an interesting observation, and we wanted to examine a little further what is the status of retinoids then in those animals. So we did a retinoid analysis where we compared the levels of all-trans-retinal, 11-cis all-trans-retinal esters, 11-cis retinal, all-trans-retinal, and all-trans-retinol, first in the dark adapted animals. And in those animals, we already saw significant accumulation of the all-trans-retinal esters, which are so the esters are actually the product of the LRAD, the direct product of the LRAD activity in this classical visual cycle. And interestingly, the same was the case when the animals were exposed to light. So no matter what light conditions we were using, we always saw accumulation of all-trans-retinal esters corresponding to the increased expression of LRAD in those animals. And we also confirmed the accumulation of esters in those animals by 2-photon imaging and looking at the fluorescence lifetime signatures that our lab previously established for those retinal esters, and the approach to image them in the retinas. So this all shows that the ester accumulation is significantly happening in those RGR deficient animals. And our explanation to this situation is that probably the retina has some kind of compensatory mechanism where in case of the RGR loss, so the loss of one of the pathways that processes all-trans-retinal, so the chromophore that is released from the opsin protein, when the photic visual cycle, so RGR-mediated visual cycle is lost, increase or overexpression of the crucial protein in the alternative pathway of utilization of all-trans-retinal is observed. And we believe that this may reflect the fact that all-trans-retinal is actually cytotoxic to cells, and all-trans-retinal needs to be, so the protein that is immediately, the chromophore that is released from the bleached opsin needs to be processed immediately to prevent damage to photoreceptor cells. And this, in our view, supports the idea that not only RGR is very important for regeneration of 11-cis retinal through this photic cycle, but also the fact itself that is picking up all-trans-retinal and it actually acts as an effective sink of all-trans-retinal plays important role in homeostasis and in maintaining the health of the mammalian retina. Yeah, this is essentially what I just mentioned. And in support of this theory, we also observed that in our study originating from our lab, where we looked at the light-sensitive ABCA4-RDH8 double knockout animals, we saw a significant upregulation of RGR in both RPE cells and Müller glia cells upon exposure to bright light. So again, it seems that RGR plays an important role in protecting the cell from damage from excessive release of all-trans-retinal or excessive accumulation of all-trans-retinal. So now back to the major point of my third aim of this project. So what is the relative contribution of RPE or of RPE originating and Müller glia originating RGR to cone function? So as I mentioned, we developed this mouse strain that features this stop cassette that is accessible by the Cre recombinase. So we crossed to study the role of RGR. We focused first on the role of RGR on cone function, and the reason was that, as I mentioned to you, RGR is a photoisomerase, so inherently it operates or it supplies 11-cis retinal in photic conditions where the cones are the major where the cones are involved in the vision, right? So to study the role of RGR on cones precisely, we turned to so-called GNAT1 mouse line that is essentially lacking rod function without affecting the overall structure of the retina or causing any retinal degeneration. And this is a well-known approach in the field. And we crossed those RGR stop and GNAT1 knockout animals with two Cre lines that were driving expression of Cre either in the RPE cells or in the Müller glia, as established and published previously. So in total, we actually generated 5 major mouse lines, which were either positive for RGR, or so wild type RGR, or RGR stop, so essentially a knockout of RGR, 2 lines that were featuring expression of CRE in RPE and in Müller glial cells, and 1 additional line that was featuring both CRE genes, so driving expression of CRE both in RPE and Müller glia. And all of those mouse lines were also crossed and bred to GNAT background to facilitate, as I mentioned, analysis only of cone function. So this is the schematic graphic of the situation or of the status of RGR expression in all those different mouse lines. Without the addition of tamoxifen, so the induction of CRE expression, our RGRs will be expressed only in so-called wild type strain, whereas upon addition of tamoxifen, we will bring back expression of RGR in RPE cells, in the RPE CRE model, in Müller glial cells, in the Müller glia model, and in both cell types in what we called double CRE line. So first, I wanted to confirm that all this genetic setup is working well in this animal model. So this is the immunohistochemical staining of the retina with anti-RGR. And as you see here in the high magnification images, we see very decent expression of RGR throughout the RPE cells. And I didn't mention, but it's important to notice that we don't see any mosaicity here, so the RGR is basically expressed in the entire layer of the RPE cells. And this is the zoom to the Müller glia area. And actually we observed, first of all, in agreement with immunostainings from other species, so from bovine, from also human, as far as I remember, the RGR was localized, curiously not to the apical processes of Müller glial cells, which are localized somewhere here in the inner segments or around the inner segments of the photoreceptors, but rather we observe expression of RGR in the end feed of those Müller glia, as shown by this staining here, for example, here, 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 and also the staining that we observed kind of confirmed what we saw previously in the single-cell RNA-seq data, so that not every single Müller glial cell is really positive for this RGR signal. So this is the status of the RGR in the wild-type animals, and here upon in this mouse line where we have the stop cassette, we see complete loss of the signal for the RGR protein. We see recovery of the signal in the RPE layer in the RPE CRE line upon addition of tamoxifen. We see recovery of the signal in the Müller glia of those animals harboring the Müller glia CRE. And we see also recovery of the signal in both cell types in the double CRE mouse line upon addition of tamoxifen. So from the genetic standpoint, all what we wanted to achieve seemed to work. And now what we decided to do is that we designed a very extensive electrophysiological experiment where we were measuring the cone sensitivity first for 1 hour in the condition where significant green background light was present and supplied to the eyes of those animals. And after this 1 hour, we were turning off the light and basically measuring the recovery of the sensitivity of cones in the darkness. So here you just see the comparison of the recordings for such an experiment for the animals wild-type animals that were either treated or not treated with tamoxifen. We didn't expect here anything to happen. We just wanted to confirm that tamoxifen doesn't have any adverse effect on those animals in the experimental setup, but that we developed here. And indeed, we see no difference in the responses of the animals treated, wild-type animals treated and not treated with tamoxifen. And here is the comparison with the knockout animals, so the stop cassette animal. And as you see, the responses were quite different from the responses of the wild-type animals. But again, tested with or without tamoxifen, we didn't see any differences between those two situations. So interesting observation here is that the loss of RGR affects both the sensitivity of cones in these photic conditions when the background light was still on, as well as the subsequent recovery of sensitivity in the darkness, where we assume that RGR protein is not efficiently working as a 11-cis retinoid, as a retinoid isomerase, trans-cis retinoid isomerase, yeah, because its activity is dependent on the light. So this was a curious observation, and we wanted to compare it with a situation. So this is just replotted responses from the previous graph. And here I compare it to responses that are actually recorded for animals that were fully dark adapted and not exposed to any background light. So here we see that dark adapted animals do not show any differences in the photosensitivity. Only the animals that were previously exposed to 1 hour of this green background light were having different responses in the darkness, suggesting in agreement with what I mentioned that RGR doesn't act directly in this dark period. However, it primes the retina to faster dark adapt when upon the transition, in the situation where you have a transition from light to darkness. These are the responses plotted again on just with dashed lines, and on this background I will show you now the responses that we recorded for all those cell lines that we developed. So we observed that both Müller glia, so recovery of expression of RGR both in RPE and Müller glia was improving the sensitivity of cones, and we also saw a clear statistically significant difference between the level of recovery of those responses upon the recovery of RGR expression in Müller glia versus the RPE cells, indicating that the pool of the RGR and the RPE, at least in mouse, is more important or has higher influence on the activity of cones in the or photosensitivity of cones in those animals. And as expected, the presence of CRE, so the recovery of expression of RGR in both cell types of RGR and so RPE and Müller glia was recovering the responses of cones to the levels recorded for the wild-type animals. Those observations were statistically significant for several points, and I'm showing you here just the significance for the points that are at the end of each of those light or dark cycles. So this is bringing me to the conclusions. So what I showed you is that photic visual cycle, which is driven by RGR, is, as we showed, sufficient to fill the gap between the RP65 mediated chromophore supply rate and the demand generated by the cone photoreceptors under sustained light conditions. We saw that Müller glia expressing RGR are transcriptionally fine-tuned to support RGR activity, and this includes, for example, approximately 1.5-fold increase in the expression of CRLBP and RDH11 and other proteins. However, the importance, of course, of those proteins on the support of photic visual cycle will need to be verified. We saw that both RPE and Müller glia are involved in the photic visual cycle, and they play non-redundant functions. So basically, if you just lose the pool of RGR from one of those cell types, you do not maintain the sensitivity of cones at the level of wild type. There's always a loss. So it means the pools are non-redundant. And the RGR contribution from RPE actually exceeds the contributions from Müller glia. However, in this case, it's worth mentioning that it likely reflects the differences in the relative RGR abundance between those two cell types, because as I mentioned to you, this RGR is much higher, is expressed at a much higher level in the RPE than in Müller glia in mouse. And RGR pools from the two cell types appeared, as I mentioned, non-redundant. And what we believe is that RGR plays a critical role in sequestering and processing all-trans-retinal in photic conditions. And this mitigates the cytotoxic potential related to the accumulation of all-trans-retinal in the retina. So with that, I want to say big thanks to my mentor, Dr. Palczewski, who spearheaded this whole project, and my collaborators, John Hong, Elliot Choi, Jennings Lu, Grażyna Palczewska, Chichen Dong, and Dominik Lewandowski were contributing to this story that I presented here today to you. David Salom, Jinya Zhang, and Huajun Yan were also contributing to other aspects of this study that were not presented here today. A big thanks to our collaborator, Dr. Kefalov, who was involved and helping us with the electrophysiology analysis of those animals, and Dr. Kolesnikov, who is a postdoc in Dr. Kefalov's lab. Thank you also to Dr. Kaiser for providing us kindly the Cree animals that we were able to use in this study, and Dr. Swarup, Brooks, and Campello from NIH, who were involved in the RNA-seq analysis of the data that I presented to you. And Robert Faris from NIH is also involved in other aspects of the study that I not presented here. And last but not least, I want to acknowledge financing from National Institutes of Health, National Eye Institute, Research to Prevent Blindness, and my support from the Knights of Templar Foundation. Thank you so much for your attention, and I'm happy to take questions.